Hello, everyone. My name is Laura, and I am joined here today with Michaela, Lillian, Bridget, and Nathan, my colleagues at Stagexa, a molecular diagnostics company. We would like to first introduce you to John. A 61-year-old father of two, he enjoys spending all of his free time with his family. John is caught off guard one morning with the sudden onset of severe headaches and dizziness. Unfortunately for John, he discovers that he has been diagnosed with glioblastoma multiforme, a very invasive and aggressive form of cancer of the brain and spinal column. Once a very active man, he now finds himself ravaged by the effects of this disease. John, his family, and his physicians now face an incredibly challenging physically and emotional battle against this disease. There are 18,000 individuals who are diagnosed each year. These patients have a devastating 3% chance of survival for the next 5 years. Despite billions spent on treatments, there's been little success. However, the recent discovery of glioblastoma subtypes has proven successful in the treatment of this disease. That is why we at Stagexa have licensed breakthrough technology from the Wistar Institute to create a novel molecular diagnostic test that can subtype glioblastoma tumors, allowing us to provide physicians with the information they need to tailor treatment protocols, improve patient outcomes. I will now turn it over to Michaela, who will address how we at Stagexa will overcome the challenges of diagnosing glioblastoma.
Thank you, Laura. The current standard of care for GBM patients hasn't really changed in the past 60 years. According to the National Comprehensive Cancer Network, this standard includes surgical debulking followed by radiation and chemotherapy. More so, only 3 new treatments have been approved by the FDA since 2005. This slow progress is in part due to GBM's complexity. Unlike the common cold, GBM tumors are heterogeneous among patients. This means that each tumor will be different depending on the patient. Current diagnostics, such as histopathology, is visual and cannot differentiate tumors adequately, thus relying on complementary molecular tests. Increasingly, molecular testing methods are being explored to improve diagnosis and prognosis. To clarify, diagnosis refers to the identification of the disease, while prognosis is a forecast of the likely outcome of the disease. As a result, research conducted by the NIH set out to classify GBM based on molecular profiles and was successful in determining 4 subtypes of GBM. However, this never translated into the clinical setting because using gene-level analysis as a diagnostic tool resulted in weak prognostic value. Thus, many doctors, like Dr. Paul Mischel, have concluded this yields limited clinical utility and don't actually use it in their practice. In an effort to change this, the Wistar Institute advanced the NIH's initiative with a patented isoform-level analysis platform. This isoform-level analysis can categorize patients into the same subtypes, but much more accurately. In fact, preliminary data has shown that these categories showcase significant differences in patient survival stratification. This has also been suggested to be due to differences in patient demographics and treatments. Thus, Stagexa seeks to demonstrate the utility of isoform-guided treatment plans for GBM patients by commercializing Wistar's technology. We provide a diagnostic service that seamlessly integrates into the traditional protocol. The biopsy sample is sent to our facility where we perform the assay, determine the subtype, and send the results to physicians. These results have increased value because Stagexa links each subtype to a specific treatment plan. For instance, even at gene level, data has shown the neural subtype has a minimal response to TMZ, an oral drug. Yet these patients are still undergoing this costly treatment. Therefore, Stagexa plans to develop isoform-guided treatment plans, IGTPs, in order to further optimize outcomes for each of the GBM subtypes. And now Lillian will discuss our plan to achieve clinical validation and adoption.
Thank you. Adoption will happen in 4 key steps, beginning with clinical validation. First, we are engaging in discussions with FDA and especially biobanks, such as those listed below, in order to perform Step 2. In Step 2, we will conduct retrospective studies, analyzing already available patient information. This is to be followed by Step 3, prospective studies to track new patient progression. With clinical data, in Step 4, Stagexa will engage with key stakeholders such as KOLs and payers anticipating market launch. Beginning with a retrospective study, Stagexa aims to analyze past patient outcomes in order to refine prognostic capability. We will acquire GBM biopsies from biobanks, subtype these samples at the isoform level, and then analyze patient data using their electronic health records. This will clarify how differently subtyped patients have responded to treatment. Next, we will conduct a prospective study to validate the use of Stagexa's isoform-guided treatment plans for GBM. This involves establishing research collaborations with neurosurgeons at large research hospitals that routinely send all tumor biopsies for further analysis. Separating GBM patients into two cohorts, those receiving traditional standard of care versus those that will receive isoform-guided treatment plans. We will then track and analyze their data for novel outcomes. Clinical studies showing improved quality of life and increased overall survival rates will inform payer, FDA, and KOLs. The core value of Stagexa is our ability to empower physicians to make better, more tailored treatment decisions for their patients. Neuro-oncologists and neurosurgeons are the end users of Stagexa's tumor profiles, and their support will be essential to our acceptance by technology review committees and insurance companies. Neuro-oncologists are key decision makers who prescribe or order tests. In a complementary role, neurosurgeons assess tumor removability, perform surgical intervention, and sometimes determine the need for additional tissue diagnostics. Based on interviews with these physicians, a technology is more likely to be adopted if it can improve diagnostic ability, predict response to therapy or survival, or aid in determining which subsequent therapies should be pursued. In order to engage with all of our key stakeholders effectively, we will be targeting journals and conferences related to neuro-oncology, such as the Journal of Nature Medicine or the Conference of Neuro-Oncology and Brain Tumor. We hope to see Stagexa within the NCCN guidelines, which will detail sequential decisions and interventions physicians follow in the United States. Lastly, given the WHO's function in setting major guidelines for brain tumor diagnosis, we anticipate the need to interact with the entity to gain support on the global stage. It is through these mediums we will drive interactions with key stakeholders such as payers. Stagexa needs to be accepted by insurance companies to ensure patient access. Diagnostics often fall under predetermined codes that health insurance companies have approved. These current procedure terminology, or CPT codes, can mean early insurance adoption, even before obtaining FDA approval, which we do anticipate as an in vitro diagnostic or medical device. The target price/CPT code associated with similar molecular diagnostics is around $3,600, and our target cost is around $140. Now I'll transition to Bridget, who will further discuss our timeline and operational strategy.
Moving on to planning for Stagexa's future, we have laid out the key financial milestones that we intend to reach within the next few years. Currently, our team is looking to start talking with the FDA to initiate the first phase of our clinical trials. Subsequently, we will be reaching out to biobanks in order to secure biopsy samples to be used in these studies. In terms of funding, our team has secured grant funding of $500,000 to begin growing out our platform, which includes establishing our laboratory facility and hiring key personnel for the lab. Our second milestone will be when we officially begin our retrospective study to validate isoform-guided treatment plans. We will be fine-tuning our technology and study in order to meet all FDA requirements. In this stage, we will be working on building up our supply chain management and seeking institutional partnerships. In order to execute these tasks, we will be initializing our Series A round of financing. In 18 months, we hope to be completed with our retrospective study and move on to our major prospective study. This will also start our Series B phase to fund the cost of our second study. As we continue, we plan to gain additional partnerships and begin our marketing efforts by utilizing the results of our first study to communicate the true value of Stagexa's test. Within 48 months, Stagexa will be completed with our prospective study. With the results from both of our studies, we hope to gain the clinical validation needed for the KOLs to facilitate adoption. Additionally, we hope to continue our effort with our marketing team and grow by establishing a new, larger lab facility. Lastly, our sales team will be up and running and preparing for market entry in 2025. In order to facilitate the milestones that we have laid out, Stagexa will be seeking $1.5 million during our Series A round. The $1.5 million will be used to fund our first retrospective study and our operations over the next 18 months. Based on our expenses, this influx will be able to give us a positive cash flow during our second year. This amount has been determined by looking at two comparable companies during their Series A round of funding. We analyzed our expenses and the deals associated with these companies to measure the exact amount needed for our Series A. As a result, we have determined that Stagexa has a pre-money valuation of $3 million by considering factors such as our licensed technology, seed money, and of course, our personnel. This valuation shows that the founders are allotting 33% of the company for this deal. Now Nathan will walk you through a little bit more about our major expenses.
Thanks, Bridget. As a diagnostic startup, we need time to validate our technology platform. And as previously mentioned, we plan to conduct 2 large-scale studies in order to do this. The $1.5 million cash influx discussed in the previous slide will be enough to take us through 2021 with cash to spare until our next round of funding. Luckily, our expenses remain lower than $2.5 million per year until 2024, so we expect our Series B round to carry us to market entry in 2025. At that time, our sales and marketing expenses will dramatically increase in order to provide ample resources to drive our test adoption. Additional major expenses include our salaries, facilities and equipment, and publication efforts. It's important to note that our sales and marketing expenses do fluctuate with our study completions, denoting the pushes we'll be making to get the word out about our technological developments. By the time we plan to launch our diagnostic test, the global molecular diagnostics market for cancer assays will balloon up to $1.42 billion. This massive increase is due to major anticipated technological innovations and is set to happen very quickly, considering the strong CAGR of this market at 10.8%. In 2024, we anticipate a 10% market penetration in terms of our target patient population, and thus a first-year revenue of approximately $6.6 million. This translates to an expected market share of a little under 0.5% for us, which is very reasonable, especially considering the dire need for a solution connecting molecular subtypes to treatment plans, which is largely echoed by the medical community. And with the strong emerging market comes the potential for a strong exit opportunity as well. We foresee a potential acquisition by a large pharmaceutical partner like Roche. Roche is the leader in oncology therapeutics and has had some major acquisitions in the molecular diagnostic space in recent years, although that's yet to happen in the context of glioblastoma. We see Roche as a parent organization that could dramatically expand our technology platform for 2 main reasons. First, Stagexa's test will enhance their cancer diagnostic portfolio, complementing the various genomic products at similar Roche-owned companies. Second, we currently have a board of directors member that is the CEO of a Roche-owned molecular diagnostics company called Foundation Medicine. Pairing these 2 key components sets us up to be a prime candidate for acquisition in 2025. We also believe Abbott Diagnostics, Cars Life Sciences, and LabCorp would be interested in an acquisition as well. Also, our team has worked hard to bring on the right biotech experts in order to successfully commercialize our technology. Cindy Peretti has joined us from Foundation Medicine. We're excited to have her expertise in commercialization and to have her on our board of directors alongside Laura and myself. We have also recruited 3 of the inventors of the Wistar technology to our advisory board, Dr. Davaluri, Dr. Sho, and Dr. O'Rourke. They possess expertise in algorithm development, assay development, and neuropathology, which will be key to the continued development of our isoform level analysis platform for additional cancer indications in the future. As a group of motivated UPenn students who are guided by such incredible experts, we believe we can change glioblastoma's standard of care for the better. We come from a variety of backgrounds, including law, product design, and bioengineering, and we've developed a strong synergy between our founders. Each of us has an intense drive to bring Stagexa's product to the market and are eager to demonstrate the utility of isoform-level gene analysis for the field of oncology. Join us in creating a new standard of care with Stagexa's glioblastoma subtyping test so we can give future patients like John a fighting chance. Thank you.